Welcome to the next session on the chapter cell cycle and cell division. There are two types of cell division, mitosis and meiosis. So let us learn about mitosis in this module. What is mitosis? It is a type of division in which the number of chromosomes in the parental cells is the same in the case of daughter cells. So there is no change in the number of chromosomes between the parental cell and the daughter cell. So such type of division in which the number of chromosomes in the parental cell and the progeny cells of the daughter cells remains the same is called as equational division. So mitosis is taking place usually in the somatic cells. What do you mean by somatic cells? Somatic cells are vegetative cells which is not involved in the process of reproduction is called as the somatic cells. So somatic cells have or these cells undergo the mitotic division. So mitosis is generally seen in diploid cells who are having those cells having two sets of chromosomes that is which is represented as 2n. So mitosis usually occur in diploid cells, but it also occurs in haploid cells of certain lower plants and also in some insects. Mitosis, a major reorganization of cell components occurs. And the mitosis, that is the division of the cell in which there is a division of nucleus and division of cytoplasm occurs, in which the division of the nucleus is called as karyokinesis can be divided into four stages. They are called as prophase, metaphase, anaphase and telophase. So once again I repeat, the division of the nucleus can be subdivided into four stages. They are prophase, metaphase, anaphase and telophase, and ultimately it results in two daughter nuclei. So what is prophase? It is the longest phase in mitosis. So of the four stages, prophase, metaphase, anaphase and telophase, the prophase constitutes the longest phase and is followed soon after the synthetic phase where the DNA undergoes replication, followed by the second gap phase or growth phase. And in the S phase and G2 phase, after the DNA replication, the DNA molecules are intertwined. They are seen in the form of chromatin reticulum. Intertwined structure is seen in the case of the DNA. So this is called as the prophase. This is the condition of the DNA in the prophase in which they are intertwined. So the nature of DNA before it enters into prophase is in an intertwined condition. So let us see what are the characteristic features which are going to take place during prophase. The chromosomal materials, which is seen in the form of chromatin reticulum, chromatin fibers, they are untangled. That means the intertwined nature, it is untangled and they condense to form the mitotic chromosome. So they take the shape of chromosomes. The intertwined chromatin reticulum or chromatin fibers, they start to unwind and take the characteristic shape of chromosomes. So they are seen to be composed of two chromatids attached together at the centromere. So each chromosome is having one centromere and two arms, and that's called as the chromatids. Studying interphase, we had learned that when the DNA is getting replicated in the nucleus, the centrosomes gets replicated in the cytoplasm. So the replicated or the doubled centrosomes, they move towards the opposite poles, okay, towards the opposite poles, and from which asters or microtubules arises. Or these asters constitute the spindle fibers which forms a mitotic apparatus which is necessary for the nucleus or the chromosomes to get separated so as to form two daughter nuclei. So during prophase, chromatin reticulum condenses to take the structure of chromosomes, and the second event which is taking place is the formation of the spindle fibers from the centrosomes. And the third event is that at the end of the prophase, the major components, that is Golgi complexes, ER, endoplasmic reticulum, nucleolus and nuclear membrane, they start to disintegrate. We can see they start to disappear at the end of prophase. So prophase comes to an end when all these Golgi complex, ER, nucleolus and nuclear membrane disappears. So after prophase, the metaphases follow. The nuclear envelope completely disappears or disintegrates. Okay, there is no presence of the nuclear membrane, The chromosomes spread throughout the cytoplasm of the cell. So this constitutes the plasma membrane. Okay, this constitutes the plasma membrane, and the whole chromosomes remain spread out in the cytoplasm. The chromosome condensation is completed, so which began in the prophase, it gets completed during the metaphase. The chromosomes or the condensed chromosomes can be easily studied, or they can be viewed under the microscope in the metaphase stage. And each has two sister chromatids. Is it clear? Each chromosome is having two sister chromatids. You can see each chromosome is having, this is one chromatid, and this is one chromatid, and this is the other chromatid. Each chromosome is showing the presence of two sister chromatids. So chromosome during the metaphase stage, they lie or they move towards the equator. Here we can see they are all distributed towards the cytoplasm. Whereas towards the middle or towards the end, what happens? They come and arrange along the equatorial plate. And this equatorial plate which is formed during metaphase is called as a metaphase plate. And they get aligned along the equatorial plate or the metaphase plate. And the spindle fibers, we can see these are the spindle fibers which is arised from the centrosomes, and we can see in each centrosome there are two centrioles which is lying perpendicular to each other, from which microtubules is arise, which gives rise to the mitotic apparatus or the spindle apparatus. And we can see over here the spindle fibers, they come and attach to the chromosomes at the centromere region. When we learned about the cell, the structure of the cell, and in that chapter we had learned about the structure of the chromosomes, and a small constriction arises on the centromere. That's a primary constriction is called as a centromere, and on the centromere a small constriction arises, and this is called as the kinetochore. So here comes the importance of kinetochore. Can you see the kinetochore in this diagram? This is the kinetochore, and in the spindle fibers which has arisen from the poles, they are attached to the chromatids by the kinetochores which is present in the centromere. So centromere has a kinetochore, and this kinetochore is a center of attachment or site of attachment of the spindle fibers. Next is anaphase. It is the shortest phase in the mitosis, and the centromere of each chromosome, they divide longitudinally. The centromere is the site where the sister chromatids, two sister chromatids are attached. So during anaphase, the centromere divides. The centromere divides longitudinally, resulting in the formation of two daughter chromatids. You can see over here, this was the structure of the chromosome in the metaphase. So here in the centromere, they divide longitudinally. See two daughter chromatids, that is the chromosomes of the future daughter nuclei. Okay, these daughter chromosomes having one chromatid each. And during this phase, the spindle fibers, they start to contract. See, we can see they can't start to move back. It is repelled back, and as a result, the daughter chromosomes, they move towards the opposite poles. So that is the main event taking place during the anaphase, that is the centromere divides, and what happens? The chromatid, the sister chromatids get separated in anaphase. So it is the shortest phase. The centromere divides, resulting in the separation of the daughter chromatids and or sister chromatids, and the sister chromatids moves towards the opposite poles, and that is the end of anaphase. After the contraction of the spindle fibers from the opposite poles, what happens? The daughter chromosomes or the separated sister chromatids moves towards the opposite poles. So they start to accumulate. Okay, the chromosomes cluster at the opposite poles, and again they uncoil. The chromosomes, they uncoil, and what happens? They forms a chromatin reticulum. You can see they form chromatin fibers. And the nuclear envelope which had disappeared during the prophase, it reappears. So the nuclear envelope assembles around the chromatin fibers, and the two daughter nuclei are formed. So the the nuclear membrane appears, the nucleolus appears, all the Golgi complex and endoplasmic reticulum reappears and the spindle fiber disappears, and we can see each cell is having two daughter nuclei, and thus it comes to an end of karyokinesis. So karyokinesis is a division of nucleus in which the nucleus undergoes division and is formed of four stages: prophase, metaphase, anaphase, and telophase. Thank you.